Let us continue our discussion on chapter 3, class 12th NCERT biology for NEET, which is the chapter is human reproduction, and the section that we are going to discuss in this video would be 3.5 fertilization and implantation. And from the beginning we are trying to understand what is explained in our NCERT. We know that NEET would be almost exclusively based on the NCERT syllabus, and our endeavor is to understand the basic concepts and the facts that are given in the NCERT. Yes, we are elaborating the concepts given in the NCERT, but ultimately what we aim to achieve is a very clear understanding of the concepts of NCERT textbook. So let us begin this topic, which is fertilization and implantation. So if you have seen the previous video, so you know that the mature Graafian follicle ruptures and releases the secondary oocyte, and that secondary oocyte is surrounded by a glycoprotein layer, which is further surrounded by a few layers of cell as well. So first let us understand what exactly was the structure that was actually released at the time of ovulation. Again, I request you that if you have any problems, please go to the previous videos, watch them, and then watch this video. So the thing that was released at the time of ovulation was the secondary oocyte, and this secondary oocyte had a haploid nucleus. This is secondary oocyte. The primary oocyte has undergone meiosis 1. This is the secondary oocyte, and this secondary oocyte has begun meiosis 2 and is itself arrested at metaphase 2. So this is secondary oocyte arrested at metaphase 2 of meiosis 2, and as I told you that this would be surrounded by a glycoprotein layer, and this glycoprotein layer would be called as the zona pellucida, and it would again be surrounded on the outside of zona pellucida by a few layers of cells, and these cells would be called as the corona radiata. So these cells, now they magically change their names. You know that in the Graafian follicle, the secondary oocyte was suspended in a mass of cell which was called as the cumulus oophorus. Now we are calling these cells as corona radiata, and obviously between the cells there would be the ground substance. So there would be the ground substance, and this ground substance would be containing hyaluronic acid. So let us label everything. So we are saying that this is secondary oocyte, and we know that this is zona pellucida or the pellucid zone. So this is the structure that actually was liberated at the time of ovulation. And please remember, I told you that what happens is that the ovulation occurs into the peritoneal cavity. So from the ovary, the ovulation occurs into the peritoneal cavity, and somehow a water current would be taking this liberated secondary oocyte towards the fallopian tube, where the finger-like projections from the infundibulum part of the fallopian tube, which are called as fimbriae, they will catch hold of the secondary oocyte surrounded by all these things and will transport it into the fallopian tube. I hope this thing is absolutely clear. So this thing is the structure that has been released. The secondary oocyte now is present in the fallopian tube, and the part that we are talking about is the infundibulum. We know that fallopian tube is lined by columnar ciliated epithelium along with peristalsis. This columnar ciliated epithelium will push the ovum towards the ampulla region. The dilated part would be called as the ampulla region, and you also know that the ovum, once released, must be fertilized within 24 hours of its release. So almost always the normal fertilization would occur in the ampullary part of the fallopian tube. The simple reason would be that 24 hours would be sufficient only to take this structure into this region of the fallopian tube at the junction between the ampulla and the isthmus, but specifically in the ampullary part of the fallopian tube. Now the oocyte is present. Now this oocyte is waiting for a sperm for fertilization to happen. So Why fertilization occurs in fallopian tube? Because fertilization must occur within 24 hours, and that is the time that is needed for the sperm, the oocyte. Sometimes I will be using the word ovum. This would be for convenience. Let me tell you right at the beginning. So when I am saying ovum, then actually I mean it is secondary oocyte that is arrested at metaphase 2 of meiosis 2, and this ovum will actually reach what is called as the ampulla, and this would be the site of fertilization, again to be specific, near the junction with the isthmus. Now I hope this thing is now absolutely clear. Now looking at the other part, because for the fertilization would be the fusion of the male and the female gamete, and we have talked about the female gamete, and this is where the female gamete is located. Now suppose the insemination occurs. Insemination is deposition of the semen in the female genital tract, and this would be a natural insemination by the sexual activity, and the semen has been deposited in the female genital tract. So we also know that semen, to begin with, is a coagulum, and after its release into the female genital tract, after some time, after say for example around 20 minutes or so, it gets fluidized, and the sperms that were entrapped in the coagulum, they are released, and then the sperms undergo a process in the female genital tract for their final maturation. So remember, during their storage in epididymis, they actually acquired the capability of fertilization and motility, but they were not fertilizing anything. The final maturation of the human sperms will occur in the female genital tract, and we are saying that okay, from the semen, the coagulum semen which gets fluidized, the sperms are released, and the sperms undergo a process which is called as capacitation. This capacitation is the final maturation. Remember, it is occurring in the female genital tract, and then as the result of the capacitation, the sperms become hypermotile. So they become vigorously motile, and you can see that the sperms do not undergo circuitous movement, they actually move something like this. So this is the type of movement that is seen in the sperms. Now they will start moving with the help of their own flagellum. Remember, the seminal plasma also contained the source of the energy, which was fructose. I hope this thing is absolutely clear. So now this semen has to cross. So the sperms have to cross what is called as the cervix. So one of the major obstacles for the sperms would be the cervix. So just to understand the things more clearly, just give me a moment. What we are saying is that this is uterus. If you remember, this is the internal os, then the cervical canal, then the external os and the vaginum, and this would be the fallopian tube. And let us say that somewhere here in this fallopian tube is the secondary oocyte surrounded by zona pellucida and some corona radiata cell, and the sperms, they have been deposited here. Now first they undergo capacitation, and then they will cross the canal, the cervical canal, and will enter into the uterus and will begin their journey towards the secondary oocyte, which is present in the ampulla, which is the site of fertilization in human beings. For that to happen, they have already undergone capacitation, they have become hypermotile, they are now vigorously motile, and at the time of ovulation, because of the presence of the large amount of estrogen, the cervix mucus also undergoes some changes and becomes passable for the sperm. So sperms can easily cross the mucus barrier that is present in the cervical canal. Again, if you want to understand the entire thing in detail, I would request you to watch the videos that we have done in our regular course for a more comprehensive discussion. But this is the final preparation for NEET entrance exam, so I am just picking up some very important points. The cervical mucus becomes passable, and the sperms cross the cervix and will reach the uterus. Not all of them, And that is why we know that in an ejaculate there may be 400 million sperm. Not all of them will be able to fertilize the oocyte. Only one sperm will be fertilizing the ovum. But for that to happen, there need to be a large number of the male gametes, and one of the problem would be crossing the cervical canal. Now this fluid, the fluid that is present in this structure surrounding the oocyte, there would be some fluid. Thus that fluid will be carrying a little bit of progesterone. So if you remember from the previous video, just before ovulation, the secretion of the progesterone, the last of the hormones to appear during the menstrual cycle actually begins, and that progesterone will help in creating the concentration gradient. A concentration gradient of certain chemicals, and attracted because of that concentration gradient, the sperms will be able, most of them actually will be able to reach the right fallopian tube. Because normally what happens that one of the ovaries alternatively release a single ovum in each cycle. So suppose in the first cycle the left ovary releases the secondary oocyte, in the next cycle the right one would do so, though it is I would say the most common occurrence, but there would be obviously variations. They are very commonly known to occur. Now because one of the fallopian tubes will be having the secondary oocyte located in it, so the sperms most of them actually must reach that particular fallopian tube, and that is because of the establishment of the concentration gradient that I was talking about. And obviously there would be some anteperistalsis, ANTE, so that means in the reverse type of direction, and that would also help the sperm, because for sperm this may be a pretty long journey. Sperm is a very tiny structure, and for sperm to reach here it must be aided by something. So it has its own flagellar movement, it is aided by the anteperistalsis, and because of the concentration gradient many of them will actually reach in the vicinity of the secondary oocyte somewhere here. So I hope this thing is absolutely clear. Now what happens is that this sperm, if you remember, at its the most anterior end, it had a cap like structure. I called this structure as an acrosome, and you know that beyond this there was head, and the head was actually having the main nuclear material or the genetic material or DNA present in the nucleus. And I told you that this acrosome contains some hydrolytic enzymes which are called as sperm lysins, and these sperm lysins will include, number one, they would be hyaluronidase. Now correlate this with the hyaluronic acid that is present in the ground substance, and then it has corona penetrating enzyme. So this is corona radiata, and finally there is zona lysine. So zona lysine, it is also called as zona lysine, also called as acrosin, and you know that this is the layer zona pellucida that we are talking about. So sperms, when they reach in the vicinity, they are having some sperm lysins in their acrosome. Obviously let us make the body as well. Now they need to penetrate these surrounding layers. They must be digested. Therefore the sperm will be able to reach the secondary oocyte and will be able to fertilize it. So just to cut the story short, you can understand that this hyaluronidase would be able to digest the hyaluronic acid present in the ground substance. The corona penetrating enzyme would be able to digest the corona radiata. So just to understand the things more clearly, let us make a few layers of corona radiata as well, and then we say that a way is carved, and because of the digestion by these enzymes, so what is going to happen is that somehow this way would be carved out, and now the sperm will be approaching the zona pellucida. When the sperm comes in the contact with the zona pellucida, there is an acrosomal reaction. So the sperms that will come in contact with zona pellucida, only they will be able to undergo acrosomal reaction. For acrosomal reaction to occur, the sperm must have underwent the sequence of events which are called as capacitation. So only capacitated sperms will undergo acrosomal reaction, and a contact with zona pellucida is important for acrosomal reaction to occur, which means that this acrosomal membrane ruptures, and this leads to the release of the zona lysine. And moreover, zona pellucida has receptors for the head of the human sperm, because now the acrosome is gone and therefore there is a docking of the head. So if you can understand, what is happening here is that this sperm is moving forward, and because now the acrosome is gone, and now the first part would be the head, followed by the neck, the body and the tail. Obviously sperm is much smaller than the oocyte. It is much smaller than the oocyte. I am drawing this diagram at a large scale. This is you do not need to draw any inference regarding the size of the relative size of the sperm and the oocyte. The oocyte will be much larger than the sperm. But just to understand this, we are saying that the zona pellucida is having receptors for the head of the sperm, and there is a sort of docking mechanism. The head of the sperm binds to the receptors that are present on the zona pellucida. Now this is species specific, which means that it is here, this is a very important role that is played by zona pellucida. It is here that it is made sure that only the human sperms will be able to fertilize the secondary oocyte. This is species specificity because of the presence of these receptors. Now what happens is that after this thing happens, now as soon as it docks onto the receptor, there are some changes that are brought about in the zona pellucida as well. So what is happening here? The capacitated sperm reaches, and then it releases some enzymes, carves away, and then it comes in contact with the zona pellucida, undergoes an acrosomal reaction. The head docks onto the receptor that are present on the zona pellucida, and simultaneously the zona pellucida undergoes what is called as a zona reaction. So zona reaction begins. This zona reaction will ultimately lead to the hardening of the zona pellucida and ablation of the remaining receptors for any other sperms that may actually try to reach it. So this is very important to understand. So that means when the sperm comes in contact with the zona pellucida, the sperm undergoes acrosomal reaction, and the zona reaction begins in the zona pellucida, leads to the hardening of zona pellucida, and ultimately to the ablation of the receptors that are present on it. So that means this zona reaction, this is again a very important function of zona pellucida, that this zona reaction, the hardening and ablation of the receptor, prevents polyspermy. Now polyspermy would be fertilization of a single ovum by more than one sperm, and obviously you can understand that if more than one sperm actually fertilize the ovum, then it will lead to polyploidy, and polyploid zygotes do not develop in human beings. So at any cost, the polyspermy has to be prevented, and a very important role is played by zona pellucida here. So second important function played by zona pellucida. Now what happens is, so let us just find some, okay, let us take another page, and now let me draw to something more realistically. This would be the secondary oocyte, and we are saying that it is surrounded by a zona pellucida, and we are saying that the sperm has already docked onto it. So let us try to get some space around it. Now again, this is highly magnified, and what has happened, that the sperm has docked onto this structure, something like this. Then what happens is that with the final vigorously motile movement of its flagellum, this part of the zona pellucida gives way, and the sperms enters into what is called as the perivitellin space. So we are not going into unnecessary detail. Now the sperm has entered here, and then will finally enter into the cytoplasm of the secondary oocyte. and that would be the apparent act of fertilization. Mind my words, that would be the apparent act of fertilization. You remember this, that this secondary oocyte would have an haploid nucleus, and the sperm will also be carrying a haploid nucleus. So the sperm, now this zona pellucida has undergone zona reaction that would prevent polyspermy. The sperm is entering into the space that is surrounding the secondary oocyte, then it enters into the secondary oocyte, which is the apparent act of fertilization, the fusion of the male and female gamete. And then what happens is that inside the secondary oocyte, the enzymes, they will hydrolyze the parts of the body of the sperm. The mid body and the tail will be dissolved, and what is left is the head part along with the neck. The proximal pair of the centriole will be released. So we know that the proximal pair of the centriole will be involved in the first division of the zygote, because this centrosome or the pair of centriole is not provided by the oocyte itself. It has to be carried by the sperm, and then after the dissolution of the membrane of the head, the haploid male nucleus is also released into the cytoplasm, and you can see a cell with two haploid nuclei. And that is why I was saying that the entry of the secondary oocyte into the cytoplasm, entry of the sperm into the cytoplasm of the secondary oocyte is an apparent act of fertilization. It is actually the fusion of the male and female gamete. Just to give you an idea of the complexity, as soon as this happens, that means as soon as the sperms enters into the cytoplasm of the secondary oocyte, the secondary oocyte, which was arrested at metaphase 2 of meiosis 2, actually completes the second meiotic division, releases the second polar body, and itself gets converted into ooted, which will be differentiated into an ovum. So this is a point to reconsider again. What I am saying is that as soon as the sperms enters into the cytoplasm, this secondary oocyte, which was arrested at metaphase 2 of meiosis 2, completes meiosis 2, releases second polar body somewhere here, and itself gets converted into ooted. The differentiated form of ooted is actually called as an ovum. And what you are seeing is a cell with two haploid nucleus, and I am not yet calling it as a zygote. I am calling it as a fertilized ovum, and this would be followed by the fusion of the two nuclei, and this will lead to the formation of a diploid nucleus with 46 chromosomes. You know that fertilization restores the diploid condition, and now this fertilized ovum would be called as zygote. So I hope this is absolutely clear. Just to give you a very quick recap, we say that secondary oocyte surrounded by zona pellucida, corona radiata and ground substance is released at the time of ovulation into the peritoneal cavity, is picked up by the fimbriae of the infundibulum, passed into the fallopian tube, and propelled by the ciliary propulsion of the columnar ciliated epithelium, and aided by the peristaltic movement, it reaches the ampullary part of the fallopian tube, which would be the site of fertilization that should occur within 24 hours of the release of the secondary oocyte. And provided the sperms are present at the same time, because there is a narrow window for the fertilization of the ovum, and the sperm can remain viable for around 72 hours in the female genital tract. This is the figure of your NCERT, though this may be up to 1 week, and therefore in the next chapter we will understand what is the rationale behind periodic abstinence as a method of natural contraception. And this insemination occurs, the coagulum dissolves, and the sperms are liberated. Sperms undergo capacitation, they cross the cervical canal, and because of the concentration gradient established with the help of progesterone surrounding the released secondary oocyte, they reach towards the fallopian tube where the secondary oocyte is located, and then the sperm carves a way into the ground substances, digesting them, the corona radiata cells, and move towards the zona pellucida. The contact with zona pellucida leads to acrosomal reaction. Then the docking of the head of the sperm onto the receptors on the zona pellucida, which is species specific. Immediately zona pellucida also begins what is called as zona reaction that will actually prevent the occurrence of the polyspermy. Then there is an apparent act of fertilization. The sperm enters into the oocyte, and as soon as that happens, the secondary oocyte completes meiosis to releases second polar body and gets converted into the ootid. The sperm that has entered into the cytoplasm will lose the midpiece and the tail, etc., and then the proximal pair of the centriole will be released that will be used in first cleavage division. Finally, the male haploid nucleus will also be released, and the thing would be called as a fertilized egg. This will be followed by the real act of the fertilization, the fusion of the two haploid nuclei, the male and the female nuclei, and this will lead to the formation of a diploid cell which is called as the zygote. And this is that cell, this is that single cell which is the beginning of the every human being. So an average human body will be containing an excess of 50 trillion cells, but all these cells will ultimately be derived from this single cell, and this fertilization has restored the normal diploid condition as well. So I hope now you understand how the fertilization is brought about and what important role does zona pellucida play here. There is another concept given in your NCERT that we have already discussed, that the sex in the human beings is actually determined. The sex of the fetus in the human beings is determined right at the time of the fertilization by the type of the sperm that is fertilizing the ovum. We have already discussed this. So what is the mechanism of sex determination in human beings? It is called as XX and XY mechanism, where male heterogamety, male is heterogametic, it has an X chromosome and a Y chromosome, and because all ova will be carrying only X chromosome, it is the type of the sperm, 50% of which will be carrying X chromosome, 50% of the sperms will be carrying the Y chromosome. It is the type of the sperm that fertilizes the ovum that decides what is the sex of the fetus. So if the egg is fertilized by a sperm which is carrying an X chromosome, the resultant sex of the fetus would be a female, and if it is fertilized by a Y chromosome, the resultant sex of the fetus would be a male. I hope you remember this. So this is the next point that is actually described by your NCERT. Now regarding the initial embryonic development leading to the implantation. So we have already discussed in the previous video that implantation in human beings occurs at the stage of what is called as blastocyst, and this occurs 7th 8th day post fertilization, and implantation actually establishes pregnancy. So mind the word, the pregnancy is established because of implantation. So it is not simply conception or the fertilization that establishes pregnancy. So this thing has to develop into an early embryonic stage. That embryonic stage is called as blastocyst. The blastocyst gets implanted onto the wall of the uterine endometrium, and if you correlate with the events in the menstrual cycle, the ovary will be having the ruptured Graafian follicle functioning as a temporary endocrine gland which is corpus luteum, and it is still secreting a large amount of progesterone that will be keeping the endometrium healthy and ready for implantation to take place. I hope now you are correlating the things that I am talking about. Now what we are saying is that, okay, so the fertilization has occurred. So the fertilization has occurred, and it has occurred in the fallopian tube. Still the thing is surrounded by zona pellucida. So this zona pellucida is a limiting type of thing that is surrounding the secondary oocyte. So normally what is going to happen, a cell grows in size. We know from class 11 cell cycle and cell division, a cell grows in size, and then to restore the normal cytoplasmic surface area, normal surface area, volume ratio and nuclear cytoplasmic ratio, it divides, and it divides by mitosis, and it leads to formation of nearly 2 equal cell replacing one cell, and this will increase, this will result in the growth of the structure, the overall size. As the number increase in normal mitosis, the overall overall growth of the structure will happen. But what is happening here is that this this zygote, which is now a diploid cell, will undergo mitosis, but this thing is surrounded by a tough zona pellucida, and till the time zona pellucida persists, the size cannot increase, the overall growth in the structure cannot occur. So whatever happens is that the divisions of the zygote and the early embryo would be essentially mitotic, but there would be a key difference. The key difference would be that there would be no overall growth of the structure, no overall size would be increased. So what is happening is that this specialized mitosis that is happening inside the zona pellucida is called as cleavage divisions, and these cleavage divisions will result in the daughter cells. These daughter cells would be called as blastomeres, and in human beings the cleavage division results in equal size of blastomere. So that means they can be unequal in certain circumstances as well, and you already know from animal kingdom that because humans are chordates and chordates are deuterostome, this cleavage is also indeterminate and radial. So in human beings the cleavage is radial, indeterminate, we know from animal kingdom. We also know that this would not lead to the overall increase in the structure, and it results in equal blastomere. So just to give you an idea, what we are saying is that this is the secondary oocyte and it is surrounded by, we say, the zona pellucida, and then there is the cleavage division. The first of the cleavage division will result in the formation of two daughter cells. So will result in the formation of two daughter cells, and these daughter cells will be formed within the confines of the zona pellucida. Just consider that the structures are roughly equal in size, and what we will see is that these are the two blastomeres. This is the equal division in the human beings, and these blastomeres, they are indeterminate, which means if they get separated somehow, each is capable of forming a complete embryo. That would be the reason of the formation of the monozygotic or identical or maternal twins. Now what happens is that this cleavage division keeps on happening, and the thing is still within the fallopian tube, and it goes on to develop into 8 to 16 cell structure, a solid ball of cells. So 8 to 16 solid balls of cells. So you can imagine, so they will be all equal. So there would be 3 cell stage, 4 cell stage, because the divisions can be asynchronous. There will not always be 2, 4, 8, 16. It can be 2 and one of them can divide early, so there would be a 3 cell stage. So these divisions can be asynchronous. This will be an again differentiating feature from the normal mitotic division, and ultimately we are seeing a mulberry type of solid ball like structure which carries around 8 to 16 cell, and this structure is called as morula. So this structure is called as morula, and then what happens is that the cells of the morula undergo a process which is called as compaction. That means they are arranged into 2 layers. Remember, it is still surrounded by the zona pellucida. There is no overall increase in the size of the overall structure, and what we are saying is that these cells, they get organized into two different layers or 2 different parts. One would be an outer cell mass. They are also called as the trophoblast cells, and the other would be called as an inner cell mass. So this thing would be called as an inner cell mass. The cells which are in green are called as inner cell mass. The light blue one, they are called as the trophoblast cells or the outer cell mass, and the thing is still surrounded by the zona pellucida. Now what happens is that these trophoblast cells, they start to secrete a fluid, and that fluid actually gets accumulated somewhere here, pushing the inner cell mass cells towards one of the poles. And what is happening here is that because of the secretion of the fluid, so let us make another structure. So don't mind an increase in the overall size, because just it is a very rough diagram that I am making. and there will be no appreciable growth in the structure. You know this. This is an outer cell mass or the trophoblast cells, and what we are saying is that because of the accumulation of the fluid, these inner cell mass cells will be pushed towards one of the poles. So this is the inner cell mass, these are the trophoblast cells, and this is the zona pellucida, and this structure would be called as the blastocyst. So this structure would be called as the blastocyst, and this cavity would be called as blastocele. So this cavity would be called as blastocele, and many R's have occurred after the fertilization of the secondary oocyte, and this thing is now coming into the uterus. So that means it is continuously traveling towards the uterus. So this was the site of the fertilization, then the cleavage division. This is happening in the fallopian tube, and then you can understand that this is entering into the cavity of the uterus. Now there are large number of cells plus there is fluid, and because the zona pellucida has been weakened by the continuous secretion of the fallopian tube as well, so it has grown weak, and because of the increased pressure from the inside, the zona pellucida gives way. It ruptures, and the blastocyst hatches out of it. So you must have seen an egg of a chicken, the chicken coming out of it. So you know that there is a crack that appears at one of the end, then the cap like structure actually goes or gives way and the chicken comes out. That is called as the hatching. The same thing happens here. What happens is that the zona pellucida actually ruptures. The zona pellucida actually ruptures, and what happens is that let us keep the color scheme correct, and let us say that these are the trophoblast cells. I hope you are getting the idea that this would be the hatching of the blastocyst, and obviously there would be some inner cell mass cells here as well. So as you can imagine that this blastocyst is coming out, and this is happening in the uterus. This is happening in the uterus, and now because the zona pellucida is gone, these trophoblast cells will come in contact with the uterine endometrium. This contact was prevented by the presence of the zona pellucida. Now this is very important. Throughout the fallopian tube, the zona pellucida persists, and the cells are not able to come in contact with the maternal tissue, and therefore no implantation occurs, and the pregnancy does not establish anywhere else but in the uterus in normal circumstances. Can you imagine what can happen if the zona pellucida goes in the fallopian tube itself? The thing will get implanted into the fallopian tube, and that would be called as an ectopic pregnancy, and the most common site of ectopic pregnancy in human being is the fallopian tube itself, and you can understand. And another important role that I have enumerated for zona pellucida. And now what happens is that, okay, let us say that this is, so let us take this is the uterine endometrium, and now what I am saying is, let me check what was the color scheme we were using. Okay, so now what I'm saying is that now the trophoblast cells, they have come in contact with the uterine endometrium because the zona pellucida is gone. And remember, now the normal mitotic type of division will occur, and the size of the structure will grow tremendously. And obviously you know that there are some inner cell mass cells here. And what happens is, we will learn in higher classes, and if you want to know the details, go to the main course video. I have explained the process in detail, that these trophoblast cells, they start to grow into the uterine endometrium. They go into the substance of the uterine endometrium, attaching this embryo onto the wall of the uterus, and this is the process which is called as implantation. And this implantation occurs 7th-8th day post fertilization at the stage of the blastocyst. So what is going to happen is, so let me tell you something. know the details in the higher class, but they start to invade into this, and what happens is that something like this structure actually is formed because of the trophoblast cell. This would be the formation of, you will know in higher classes, what is called as chorion, and this is one of the extra embryonic membrane. So that means the trophoblast takes part in the formation of placenta, and this structure along with allantois forms the chorioallantoic placenta in the human beings. And this trophoblast cell, now there is a problem here. The problem is it is 7th 8th day post fertilization, and this is the time when corpus luteum starts degeneration. And we do not want that the corpus luteum should be degenerated because fertilization has occurred. And if corpus luteum gets degenerated, it will stop the production of progesterone, the support of the endometrium will be withdrawn, and there would be the sloughing of the wall of the uterus or the endometrium that will lead to the menstrual flow, and obviously along with it the established pregnancy, and that would be a miscarriage or the wastage of the pregnancy. That should not happen. Now, in order to corpus luteum should function, there must be present LH, and LH is not being secreted because of the presence of progesterone itself. So this is a problem. And what is LH? LH is a gonadotropin. So we need a gonadotropin which will have some function similar to the LH, but should not be under the negative inhibition of the progesterone itself, and this cannot come from the mother body. And therefore you can understand these trophoblast cells also secrete a very important hormone, and this hormone is called as the human chorionic gonadotropin. I have already told you that this membrane would be called as chorion. This is because of the trophoblast cell, and now you know that this thing is called as HCG, human chorionic gonadotropin. For God's sake, try to understand that this actually is a gonadotropin. It is similar to LH, but is not LH. It is coming from the fetus side. It goes into the mother body and maintains corpus luteum. And if the corpus luteum is maintained, there will be no menstrual flow. And I told you that the absence of the menstrual flow on the expected date would be the first clinical sign of the pregnancy, because menstrual flow will not occur. And this is a hormone that is actually produced from the fetal cells. And this goes into the mother body. Mother will eliminate HCG in her urine. Mother is eliminating HCG in her urine, and because HCG can only be present if implantation has occurred, detection of HCG in the urine of this female will be a test for the confirmation of pregnancy, the pregnancy test. So in the pregnancy test, we detect the presence of the HCG in the urine of the pregnant female, and because we know that the HCG is not produced by the mother, a non-pregnant female, it would be produced by the fetal cell. Therefore, this would be indicative of a pregnancy being established. I hope you got the idea. And now what is happening, because progesterone is still there, corpus luteum has been maintained, so progesterone is there, estrogen is there, and the endometrium actually keeps on flourishing. So endometrium keeps on flourishing. Ultimately, it will be surrounding the developing embryo on all the sides, and incidentally, after implantation, the endometrium will now magically be called as a decidua, and some of the maternal tissues and the fetal tissues will lead to the formation of ultimately what is called as placenta, will lead to the formation of placenta, and the embryo proper will be formed from the inner cell mass. So the embryo proper would be formed from the inner cell mass. The trophoblast cells take part in the formation of the placenta, and this placenta establishes a link between the mother body and the fetus as well. So I hope this thing is now absolutely clear, and this is what has been discussed in this section of your NCERT. So this is fertilization, the early embryonic development leading to the implantation and leading to establishment of the pregnancy. Now we have an idea that what is happening. The inner cell mass will be forming the embryo proper, and the placenta would be formed by the trophoblast cells. So we will be stopping here. When I come back, we will discuss the remaining portion of this chapter in the final video on our discussion on chapter 3.